Alrighty, everybody, welcome to Lecture 10. This is a very long lecture, apparently. So I don't want to take up too much of people's time, so I'm going to try and go through it kind of quickly. I don't think all of it is that important, so we'll do our best to go through the important bits. But yeah, at the time of recording this, you guys have finished your first exam, so congratulations. I will be uploading resources for exam 2 sooner rather than later, so be on the lookout for that. Check the IC Canvas page for Biochem 442. And yeah, I mean, if you're watching this from the IC, that is. If you're not, then cool. But yeah, let's go ahead and get into this. So lecture 10 is going to be looking at things like actin and cell structure. So let's go ahead and jump right into it. So real quick to start off, what are the functions of the cytoskeleton? It has a lot of different functions other than cell structure. It helps with transport and a whole bunch of other things. So you can see here, intracellular transport, cell movement, internal movement of cell, like resistance to pressure, and then cell shape, of course. The neurons are very highly dependent on things like microtubule for their cell shapes. So what are the three main families of protein filaments? And protein filaments is just, things that make up the structure of a cell. So we've got a couple of them. There's going to be microtubules, which are like the big boys. Actin, which is going to be the small boys, and then intermediate filaments like lamins and keratins, which are going to be the medium boys. So we think of these as microtubules as one category, microfilaments, which is basically actin, and intermediate filaments, which makes up all of these other things: keratin, desmins, lamins, vimentins, and nestins, which are things they've actually already listed here, of course. So let's take a look at an example of the cytoskeleton in action, and how it works in muscles. So to start off, there's three types of muscles. There are skeletal muscles, cardiac muscles, and smooth muscles. Here they are. So skeletal, cardiac, and smooth. And the thing to keep in mind is where they're located and the striations, if you will. So the skeletal muscles are things like in your legs, your biceps, all that stuff. Heart muscles are obviously in the heart, and then your smooth muscles are muscles that make up your internal organs. So things like your liver, your stomach's probably a better example. Your stomach is a great example of and your intestines are great examples of organs that have smooth muscles. So those are not really striated, if you will. So smooth muscles don't actually have these striations, so that's what kind of makes smooth muscle a bit different from skeletal muscle and cardiac muscle. So if you guys have already taken Bio 220, hopefully you remember a decent amount of this, or any sort of intro bio course that's covered muscle contraction and neuronal firing in that sense. So muscle structure is going to be, we've got a skeletal muscle, which is made up of many different muscle fibers, and a muscle fiber is made up of hundreds of thousands of myofibrils. And so that's kind of the cool thing, or the important thing to keep in mind, is that there's hundreds of thousands of these within one muscle fiber, and then there's thousands of these within one skeletal muscle. So your skeletal muscles are, oh my goodness, very large in terms of how many myofibrils are in there. So, and they all have to be coordinated in unison, right, to commit to an actual contractile force. So, yeah. This is an example of what would, if you were to zoom into this portion here, like if you see these like lines here, this is what it looks like inside. So, yeah. So the cool thing about muscle contraction is it involves it involves actin. Myosin is not one of the different cytoskeletal proteins, but it involves actin, and actin is the microfilament. Remember, so actin has two ends. It has a plus end and a minus end, and we'll talk about why that's important in a second. What you should keep in mind for this is, this kind of structure here. We've got our actin filaments that are on the edges, and our myosin's kind of in the middle. And remember, the myosin has these myosin heads that pull the actin inwards. So if you can imagine, if you pull this inwards, it makes this boundary smaller, and so that's what we call contraction. And relaxation would be the opposite. Relaxation would just be when these are allowed to move outward instead. So here we've got a example of what myosin looks like. So myosin is made up of these protein things that wrap around each other. We've got our C termini. And the important thing to keep in mind is the light chains are these portions here that kind of hold the heavy chains, which are the heads, to these coils. So we've got two alpha helices. Alpha helices, remember, are protein structures. We've got a light chain coming off of either side of this alpha helix, and then we have our heavy chains. So these are two different alpha helices that are coiled around each other. So the C terminus is here, and the N terminus is always at the end. So they're two different alpha helices, right? Two different protein products that have a N-terminal heavy chain and a C-terminal alpha helix. And so when you wrap them around each other, you get these myosin heads. And these myosin heads are the ones that, when they move, cause contraction alongside actin. So a couple of things to keep in mind about the muscle structure and muscle cells. Firstly, the M line. So the M line is just the middle. It's the middle of the myosin specifically. So, yeah. Just as a bit of a reminder, this section here that we see is actually called the sarcomere. So remember, myofibril is this everything here. The sarcomere is specifically this section that we're looking at. So yeah, I don't think that's too important, to be honest with you. A lot of these individual things are kind of meh in terms of how important they are to know, but they could show up on a quiz, so which is why it's important to actually at least study them a little bit. So the things to keep in mind are the M line is the middle. The Z line is this section here where it kind of looks like a Z, if you will. It's kind of hard to describe, but you can see like this as like a Z. So there's the Z line is like the end here, where we've got like this Z-ish pattern going on. So I like to think of it as like Z like this. So that's the Z line. It's at the end of the plus ends for the actin filaments. We also have the this whole idea of sliding filament theory, by the way, is just this idea that these things shift. What they're showing here is like it should be an expansion, basically. So sliding is just that these heads basically pull the actin like a rope, basically, is the idea. They slide along the actin and they pull on it. I think that's what they're trying to get across with that. So yeah, here you can see this, right? So you can see that when the myosin kind of slides and pulls this way, everything gets smaller, and so everything contracts and compacts together. So now let's talk about the process of contraction. What you should keep in mind is, first you have to send an excitatory signal from a neuron to trigger calcium release, which is what causes contraction. So basically, the first thing we have to do is we have to send an action potential down a motor neuron. So we have our brain sends a signal to our neurons, our motor neurons, and those synapse onto our muscles, and that leads to muscular action potentials. So excitatory signal from neuron triggers calcium. So once that neuron synapses onto a myofibril or a sarcomere, what that does is it releases acetylcholine, and the acetylcholine binds to some receptors, which leads to an action potential in the muscle itself. So what's cool about this is this section here, remember, we call it a synaptic cleft when it's between two neurons. when we are doing it between a neuron and a muscle, we call it the neuromuscular junction, or the NMJ. And so that's a cool little thing to remember about that. And so once these acetylcholine molecules bind to these receptors, it causes sodium influx into the cell, which we call depolarization. And so that sodium basically travels, and it basically travels this way, and it eventually makes its way into these structures called T-tubules. So it doesn't go into the T-tubule. I want to be very clear about that. The T-tubule is like an indent. So the sodium itself doesn't go into the T-tubule, but it does kind of travel along this wall, if you will. So it kind of travels like this. And so it depolarizes this entire area. And the T-tubule stands for the transverse tubule. So this whole section here, right, this kind of indent here, is the T-tubule. And so that T-tubule has a direct connection to the sarcoplasmic reticulum, which is this structure here. And basically, when the depolarization happens, it causes a door to open on the sarcoplasmic reticulum that releases calcium. I wonder if they show that here. They don't show the door specifically, so I don't think it's that important to know about that door. What we should know is that once this T-tubule depolarizes, it's like physically connected to the sarcoplasmic reticulum, and so that door, when it gets depolarized, opens the door, and then all the calcium flows out of the SR into the muscle itself, into the muscle cell itself. And so now this is where we can talk about the effect of the calcium. Like, what does the calcium do? So calcium is important in this because calcium is actually what initiates the muscle contraction process. So calcium first has to bind to a protein complex called troponin. Troponin is made up of 3 different proteins, troponin I, C, and T. I personally didn't think that these are super important to remember. It might be something you jot on your note card in like a couple words, but it's not something that you should like harp on for a super long time, I don't think. But a couple other things to keep in mind is troponin. This is where the calcium binds. So the blue is the calcium. But troponin is attached to... Oh, don't do that. Troponin is attached to tropomyosin. And tropomyosin is attached to the myosin itself. Not myosin, sorry, the actin itself. So essentially what we're going to do is... did I say myosin? I don't know. I'll explain it better. But so basically the idea here is calcium will bind to troponin, right? And then after calcium binds troponin, troponin kind of shifts out of the way of tropomyosin. So troponin kind of not leaves, but it like shifts away slightly, and it actually reveals a binding site on tropomyosin for the myosin head to bind. So basically, calcium binds to troponin, troponin shifts out of the way, and then and then the tropomyosin has a binding slot on it that the what's it called? The myosin can bind to. So actually, the way that they've drawn it here is you see this like red section here. See how it's covered by the purple tropomyosin? What happens is when this calcium binds, the blue binds, it causes a shift, so this purple section moves up. See how this is moved up now? And so now these red spots are free for the myosin to bind to it. And so the myosin will bind to it. And yeah, and then once the myosin binds to it, you can use ATP hydrolysis to start pulling on it. So here is just a nice overarching diagram of what is happening. This is probably the one that you want to put on your note card or just remember. It's very helpful. So start with our neuron, send an action potential to our muscle cell. The motor neuron sends acetylcholine to the neuromuscular junction, which causes acetylcholine binding to these receptors. That opens the sodium channels so that sodium fluxes in. And that sodium current / action potential travels down the muscle cell into the T-tubule area, which is directly connected to the sarcoplasmic reticulum. And so after that area is depolarized, the sarcoplasmic reticulum opens a physical door, which allows calcium to flow into the sarcoplasm. And the sarcoplasm, by the way, is just the cytoplasm of a muscle cell. And so once that happens, the calcium binds to troponin, which causes tropomyosin to shift out of the way. And then myosin is able to bind to actin and cause contraction. Alright, a closer look at troponin tropomyosin, right? So here we can take a little bit of a look at what is happening in a bit more depth. So, again, this is probably where I'd say it's important to know these three. I think they're relatively simple to remember, so, I mean, they're named pretty coherently. So I is inhibitory calcium, or C is calcium binding, and T is tropomyosin binding. And ideally, you can probably tell what's going on here. So, yeah, let's see here. Actin, they don't really show it too well in this, but, yeah, basically you can see this, like, these blue sections are where the troponin, the purple, binds. Or sorry, the tropomyosin binds. The C is where the calcium binds, and the I is just kind of this inhibitory complex that stops anything from binding in the first place, or stops the myosin from binding, if I'm not mistaken. I might need to correct that, but later on. But yeah, this I don't think again is that important to keep in mind. They show it in more depth down here, so we can talk about it later a bit more. So what molecule does it represent? Calmodulin. Hopefully you remember calmodulin and calmodulin kinase. Remember, it binds 4 different calciums and does all that. So normally troponin and actin are bound in this way. When you bind calcium to troponin, though, the troponin itself actually shifts out of the way. So yeah, we can see that here. Troponin, no signal. Troponin holds tropomyosin in place to block the myosin binding domains. They actually should have shown it a bit better here. It's actually, troponin holds tropomyosin, so there actually should be a myosin somewhere here, but it's not shown. But then once troponin binds calcium, the troponin pulls the tropomyosin away, which is what reveals the binding site on the actin. So the signal is, calcium. So calcium causes troponin to release, allowing tropomyosin to move out of the way. And that allows myosin binding to the actin directly. So yeah, here's a closer look. This is what I actually care about. So we can see here we have our calcium binding, our tropomyosin binding, and our inhibitory area for this complex. You can see here the calcium binding, right? So normally it's under this configuration. When there's no signal, you can see that the troponin, or the T region, is holding on to this the tropomyosin. So troponin holds tropomyosin in place to block the myosin binding, and the inhibitory region kind of assists with that. So the troponin is binding to the tropomyosin, or the T region is binding to tropomyosin, and the inhibitory region is kind of binding to the actin to stop it from really moving around. When calcium binds to the calcium binding region, the inhibitory region shifts upwards. Actually, probably should use a different color. Shifts upwards. Not a better color I could use? That's fine, whatever. I'll use purple maybe. Shifts upwards, and that causes the and then at the same time the T region kind of shifts downwards a little bit, and pulls this up with it. It pulls this purple tropomyosin up with it. So calcium binds, everything shifts. Calcium causes troponin to release, which allows the tropomyosin to move out of the way and permit myosin binding. So this basically moves upwards, is what's going on here. So kind of to summarize,
the calcium binding region troponin binds the calcium, which causes the inhibitory region to release. And so once the inhibitory region has released, the purple region can move up. The purple tropomyosin can move up. That's the really important part of it. Because like the tropomyosin binding, all this is really doing is it's keeping the troponin complex stuck to the tropomyosin. The inhibitory complex is the one that kind of stops the tropomyosin on the bottom here from shifting upwards. So once this is moved out of the way by shifting upwards, then the bottom region can shift upwards and it's okay. Okay. Yeah, hopefully that makes sense. I hope I'm not doing too fast. Again, feel free to leave comments and questions if you need me to answer them. I'll take a look at them. Yeah, I'm trying to think if there's anything else I want to mention here, but yeah, again, I just want to make it very clear what I'm trying to get at here. The troponin, the T portion, all this is doing is it's keeping the troponin complex tethered to the top portion of tropomyosin. The calcium binding region just binds calcium, which is what initiates the conformational change. And the inhibitory region is the main one that is stopping this bottom tropomyosin from shifting around all the time. So once this inhibitory tropomyosin complex moves upwards, then the tropomyosin down here can shift. That's the main thing that I want to keep in mind there. Okay, so this is just a cool little piece of info. Cardiac troponin tests are used to assess heart attacks. So, one way to determine if a person has had a heart attack is actually to take take a sample and look for presence of cardiac troponins. Because when a heart attack happens, those tissues that tissue becomes ischemic, so it actually loses blood flow, and so it actually causes bursting. And so if you were to look at the bloodstream and you were to see just floating troponins in your bloodstream, then it's a sign that the person has actually probably had a heart attack before. So the idea there is basically, again, heart attack, the heart is ischemic, the heart doesn't have oxygen, and so when the heart doesn't have oxygen, some of those cells actually end up bursting. And so when they burst, they release their contents into the blood, and so we can actually measure troponin content to see if there has been any bursting. Because again, troponin shouldn't just show up in the blood. So if the heart cells have bursted, they release their troponin into the blood. You can track that troponin, and you can tell that they've had a heart attack. Yeah. So now we can actually get to talk about how myosin works a little bit. So again, we talked about actin. Actin has a binding site for myosin on it, and the myosin head specifically. So let's start with this. So we're going to start with this. So this is currently the relaxed state. Nothing has happened. We have our actin's chilling, our myosin's not doing anything, it's just sitting there. I wish they showed images directly. Yes, here we go. Okay, so they don't really do a great job of showing what is happening here. Yeah, so I guess I'll just go through these one by one. So this is the relaxed state. Basically, at this point, what has happened is we used to have an ATP here, but that ATP got hydrolyzed, and now we have an ADP and PI. That is what's happening. Nothing is released, and so basically the hydrolysis energy from taking this ATP and breaking it is kind of stored for a power stroke, when the power stroke is the part where we actually shift this. But in this state right here, when it's ADP and PI bound, the myosin head has actually a pretty weak affinity for actin, so it actually doesn't bind. And now we move on to this. So once, we've hydrolyzed this ATP, we have our ADP and PI. It's weak affinity, it's not going to bind. Then we release our inorganic phosphate. So that's one of the worst things I've drawn. This kind of leaves. And so now, with just ADP bound, now we have a very strong affinity for the actin. And so what happens is the ADP kind of myosin head binds to the actin binding site, or the myosin binding site on the actin, and causes a power stroke. So it pulls everything this way. So this head kind of shifts this way and pulls the entire actin with it. Like this. And when it does that, the ADP is also released at the same time, basically. So this phosphate release is used to bind to the actin itself, and then the power stroke happens, and the ADP is released. And then a new ATP comes in and binds to the myosin head, which and then the hydrolysis happens. So the ATP binds, this gets hydrolyzed, and that pushes that one unbinds the myosin from the actin, and two, it pulls the myosin head back to the original starting position. And now we're back here. So we're at the hydrolysis portion. So it's been hydrolyzed, right? So now we have this ADP and PI. Then again, we're going to repeat the process. The PI is going to leave. The ADP myosin head will bind to the myosin binding sites. It will cause the power stroke. The ADP will be released. Then the ATP will bind and then hydrolyze to cock this back into this position again, and you reset. So here is a great diagram of what this looks like. So here we can see right now we have an attached myosin head. You're going to add an ATP. The ATP is going to disconnect the myosin from the actin. Hydrolyzing that ATP causes this the myosin to shift back to its original position. And so now what we do is we release the phosphate. That causes the strength of the binding between the myosin and the actin to increase, so they bind. And then the power stroke happens and ADP is released. And so now we are back at this step up here where we need to bind ATP, hydrolyze him to put him back into this spot. And then we do the power stroke. And then we're back here again. So that's the main thing to keep in mind for that. All this is trying to say is that there is a disease where the septum of the heart, which is like the middle lining, has a larger muscle mass and actually causes impaired ability to pump blood, or just impaired ability to pump blood is the main idea. Some cool videos, actually, that I would recommend taking a look at if you're interested in them. And so now we take a look at a little bit more to do with these. I do wish they showed a little bit more of that, but that's okay. So cytoskeleton action, dynamic action, dynamic actin, sorry. So now we're going to talk about actin in a bit of a different context. So actin filaments are comprised of monomers with an ATP core. So essentially what that means is there's basically actin, actin protein, and there's going to be like some ATP molecule inside, or it's going to be attached somewhere to the end or something like that. What's important about this is that the stability of an actin polymer is actually dependent on whether or not ATP is present or not. So one thing that we need to keep in mind is we have this idea of fast growing and fast shrinking. Fast growing and slow growing, and that also corresponds to slow growing and fast shrinking. Slow shrinking and fast shrinking. Gosh. Anyways, so the plus end of any actin filament grows fast and also shrinks fast, while the minus end of any actin grows slowly and also shrinks slowly. So what we should look at here is, with this actin ATP kind of form, when ATP is bound to actin, the actin monomer has a high affinity for other actins, so actin monomers. But when the ATP is hydrolyzed to ADP, the actin has a lower affinity for other actin molecules. So this is actually a really bizarre image. I wish it was drawn a bit better. That's okay. Basically, the idea here is that in this ATP form, it's gonna bind to this other actin because it's very highly has a very high affinity for it. So very high affinity. It has the ATP. It's gonna the monomer is gonna bind to the other actin monomers to make this polymer. These what's it called? These ADP versions have a much lower affinity for each other, though, actually. So they're actually going to start slowly disassociating and breaking off. It's a bit of a different idea there. So I like how they show this. I wish they I needed to show. Yeah, here. So let's just look at this like that. So essentially, these are all going to be ATP bound subunits. They're all going to combine, and then eventually you have enough of them where you start to enough ATP forms where they start to bind to each other, and you make this massive filament. And so as this is happening, the actin monomers are in a process of hydrolyzing ATP or not hydrolyzing their ATP. So there's going to be some versions that have ATP bound and other versions that don't have ATP bound and have ADP bound instead. And so there's like a constant thing of it's growing and it's shrinking at the same time. So it seems like it's staying the same size, or it might seem like it's growing. So it kind of depends on which side is growing, which side is shrinking. So the important thing to keep in mind with these is there's a couple different phases. So the nucleation lag phase is kind of when we're kind of waiting for some of these actin monomers to combine together. And then when we start making oligomers, this is where the growth phase really starts to happen, where as this gets larger, the probability of that oligomer finding other oligomers and finding other actin molecules becomes much larger. And so you get this exponential curve where you eventually start making massive structures. And at some point you get to the point where you're at a steady state phase where the rate of filaments coming off is equal to the rate of the rate of actin coming on is equal and opposite to the rate of actin coming off. So that's kind of the cool thing about actin. So this is just a really interesting slide. All it wants to show you is that actin has a lot of different roles in cell structure. We talked about actin specifically in muscle cells and how it's involved in that cross-bridge cycling process with the myosin head, but actin's involved in a whole bunch of other things. So it's involved in things like shown here. These are all structural cell things. So remember, in muscle cells it would be used for contraction, which is like a cellular function, but here it's just used for more like traditional structural things. So like stress fibers here, it makes up stress fibers. Stress fibers are how the cell kind of resists pressure, if you will. There's also cell cortex. This is kind of how it helps organize the cellular compartments and things like that. Dendritic networks and filopodiums. These again help make protrusions, and maybe these protrusions are helpful in certain things. Remember when you guys did Sonic the Hedgehog stuff, you guys needed like that protrusion, that microvilli to occur in order for anything to happen in that signaling pathway. Microtubules could help with forming those kind of microvilli. So that's kind of the cool thing about microvilli and about actin in general, is that it's very involved in a lot of these processes. So here are some more
examples of what it does. You get cases where in non-muscle cells, maybe they bind parallel to each other. Maybe they bind to actinin, and that maybe they kind of contract in this way maybe instead. So there's a whole bunch of different ways that you can use actin, basically, is what it's trying to say. But this is one of the things I would like to cover, is actin-mediated cell motility. Basically, this is a really interesting kind of process, because this is how cells move when they're not floating in a liquid, specifically. So, steps in actin-mediated cell motility. First, we have to, look at how it's attached in the first place. So first, the cell is going to attach to any given surface with these focal adhesions. These are parts of the cell itself. They kind of form when the cell is trying to attach to something, and they involve actin in them. So we can see here, these are attached here with these focal adhesions. And what happens after this is we send out a lamellipodia, and that is similar to a philopodium, but it's here like these lamellipodium. So these lamellipodium are sent out, and so basically this has to happen through growing of an actin filament, right? So an actin filament has to grow from its plus end to extend forward so that it can form this lamellipodium. And once this lamellipodium is large enough, it'll form a new adhesion section onto the surface that it's on. Then the cell actually uses a myosin kind of mechanism to uses the myosin heads to pull the cell in the direction of movement. And so what happens is that causes a kind of severing and a disconnect between the old adhesion, or the adhesion that was initially there, and the actin, and so now the cell has shifted. Another cool example of actin in progress, or in the process of doing its job, is dystrophy, and a muscular dystrophy, or Duchenne's muscular dystrophy, involves a protein called dystrophin. It's actually the largest gene in the human genome, if I'm not mistaken. It's incredibly large. So yeah, it actually causes degeneration of a lot of muscle fibers. So for cells that aren't muscle cells, normally this is what actin kind of looks like. Attached to the membrane of any given cell, there's a capping protein, and there's like some membrane proteins and anchoring proteins that kind of stop the actin from degrading that much. And so then you have the actin here. And what makes this process very different in muscle cells is that instead of attaching the anchoring proteins and only having one actin molecule, or one actin polymer per capping protein, or per blue anchoring protein, basal lamina, if you will, instead what happens is each of these basal lamina attached dystroglycans, these blue guys here, attached to a single dystrophin protein. But that single dystrophin protein is absolutely massive. It's like a massive tray. And so you can think of it like what's a good example of this? Like, maybe like you can think of it as a tray that stores cups on it, or something like that. Like a tray with hundreds of different cup holders, right? So in non-muscle cells, every cup, there's only one cup holder for this actin. So that actin is only going to fit into this cup holder, and you can only have one actin in one cup holder in this case. In this case, we have a big tray of cup holders. And so instead of just having one cup with one actin, you now have a massive set of cup holders where you can put a bunch of different actins in. So a bunch of actins can bind here. You can have an actin here, an actin here, an actin here, an actin here. So basically what I'm trying to get at here is dystrophin allows you to have way more actins attached to one dystroglycan than just a normal capping protein setup. And so why this is important is it's important because it's what allows your muscles to generate so much force. It allows your muscles to generate so much force. Yeah. I'm trying to see if there's anything else I want to mention here. This is a much stronger attachment to the ECM. Yeah, all they're trying to say here is that this laminin attachment is a stronger attachment than this, and it allows it to bear all the forces without ripping. So the laminin is a little bit more flexible, right? Laminin is actually one of the intermediate filaments, if you remember right, if I'm not mistaken. And so the laminin basically is a little bit more flexible than just attaching the basal lamina directly to the dystroglycan. And so that's what allows it to bear so much more force. On top of the fact that, again, the dystrophin allows for a lot of actin storage, which is what allows it to generate so much force in the first place. It's basically like in this, you're basically maximizing the actins per unit area that you can carry, which is why it's so helpful. Now we can talk about microtubules. I wish they kind of showed what they were talking about. I think this one is like, yeah, I mean, it's fine. Yeah, we're talking about microtubules a bit. So microtubules, microtubules are cool. So actin, right, uses ATP. Microtubules use GTP. And there are two types of monomer in the microtubule space. It is the beta tubulin and alpha tubulin. So here we have a quote-unquote monomer of microtubules. Is actually a dimer, so it's actually a heterodimer. So a single unit in a microtubule is a heterodimer of an alpha and a beta tubulin combined. The plus end of the microtubule is always the one that has the beta dimer at the end of it, and the minus end is the one that has the alpha tubulin at the end of it. So you can see here, like, this right here is going to be the beta tubulin end, so that's going to be the plus end of the microtubule. That's going to be the growing end of it. And so it's important to know is that we have GTP binding to both the beta and alpha tubulin, if I'm not mistaken. And, or actually, I can't actually remember. Does it bind to both? I can't remember. I believe it actually only binds to one end. But we'll take a look at it in a second anyways. Here's some cool images of actually what they look like under electron microscopy. It's actually really, really interesting. These structures are so wild to look at. But yeah. Anyways, a cool thing to keep in mind about microtubules is they kind of form these like cylindrical structures. So unlike actin, which forms these helical structures, microtubules are actually hollow in the middle pretty much. So they form like these lines in rows, and they basically form a cylinder from these dimers, which is pretty cool. So there are a couple different names that we should keep in mind for how microtubules work and how they grow and shrink. So the way that it works is the GTP cap is what allows for stable growth of the microtubules. So when we have our GTP on the end of our beta tubulin, we can attach more of these heterodimers to each other, right? So you can see here rapid growth with the GTP cap end. So the growth occurs when the GTP is attached, which is what the square represents. The square represents with the GTP. So it's going to keep growing when the GTP is attached. Randomly, when the GTP is hydrolyzed, or maybe the GTP falls off, right, or after a certain amount of time, when the GTP has hydrolyzed itself, it causes rapid shrinking, and that is called the process of catastrophe. So catastrophe is this idea of breaking down, if you want to think of it like that. So catastrophe is breaking down, and so that's when the GTP has fallen off or has been hydrolyzed, and then so it starts to shrink. Again, through some random process, the GTP cap will be regained, so that that heterodimer will bind a GTP again to its beta subunit, most likely, or beta tubulin
unit, most likely, and then the growth will continue again. And that process repeats. So basically, you gain GTP, you grow, you lose GTP, you shrink. Gain, grow, lose, shrink. And so here is an image of what that looks like. You can see the red is meant to be the ATPs, or GTPs, sorry. So you can see how these regions are much more stable, and so it's going to keep growing. But these regions are not as stable, so they actually could undergo a catastrophe kind of process. But usually it grows and shrinks from one end. It usually grows and shrinks from this top end here. I want to double check something actually. Yeah, that's something to keep in mind, yeah. So, again, microtubules, they mainly only grow and shrink from the plus end. That's the main thing to remember. So they don't really shrink from the minus end that much, or grow at all from the minus end. And mostly it's from the plus end. So that's why you can see here that's growing from this end and catastrophizing from this end. That's different from actin, because actin tends to grow from the plus end and shrink from the minus end, if I remember correctly. Yeah. So remember, so that's a bit different from actin. So actin can grow and shrink from both ends, but it grows and shrinks faster at the plus end than the minus end. Microtubules, on the other hand, they pretty much just grow and shrink from the plus end only. Here's a really cool video of what it looks like for microtubules. I actually would recommend watching that. This is just looking at what it looks like in vivo, so we can actually watch cells use microtubules. Pretty fun. So what's the role of microtubules? So microtubules actually serve as one of the most important structural proteins in vesicular transport. So neurons rely heavily on microtubules because neurons make acetylcholine and all of their neurotransmitters much closer to the nucleus, and those vesicles have to be shuttled all the way to the axon terminal. And neurons, remember, can be like up to a couple meters long, if I'm not mistaken. So they are huge. So yeah, here's a cool little image or kind of thought experiment. Vesicles are trafficked insanely fast. So if you were to scale it up to the size of a car, it would drive 6,000 miles in a day, which is 350 miles per hour. So just remember, these microscopic processes occur at extremely fast rates. They don't occur on the scale of like seconds, minutes, or hours. They're occurring on the scale of like microseconds, milliseconds, nanoseconds. They're quick. So, yeah. So dyneins and kinesins, and I have a bit of a trick for how I remember this. Dyneins and kinesins are trafficking proteins. They are actually very similar to how myosin works, but basically they help traffic vesicles. They basically carry vesicles on their back. Please watch this video if you haven't watched it. There's only so much I can do to explain how kinesin and dynein work. This video does a very good job of explaining how it works, so please watch them. So dyneins and kinesins, basically what's happening is they're ATP-dependent microtubule motor proteins, so they aren't made of microtubules, but they only walk properly on microtubules, really. There are always exceptions, but they generally only work on microtubules. So it's basically got two legs and a body, and the body connects directly to a vesicle that it's carrying. So you can imagine that it's carrying a big vesicle up here, and then these legs are basically walking. So what happens is you're going to have ATP bound to one leg and ADP bound to the other. And so what happens is the ATP is hydrolyzed to ADP and PI. And then that hydrolyzation causes this leg to shift out in front. And then while that is happening, an ATP binds the leg that is in the back. and then that process repeats. The ATP is hydrolyzed, this leg shifts out in front, this leg binds another ATP, and then you continue the process all over again. So as you know, that process is how these things move along the microtubules. And now, these things, remember, are moving at 350 miles per hour. So they are moving fast. Very, very fast. You can't even see how fast they're moving. These are things that you must memorize. I'm going to be honest with you. Taxol, colchicine, I keep messing this one on. Colcimide is good. Vincristin and nicotazole is the other one. So you got to memorize these. I think colchicine, I think is how you're supposed to say it. I can't remember how to say it. But yeah, this, vincristin, and then nicotazole. So yeah, basically what this does is, you remember, microtubules polymerize from their plus end and depolymerize from their plus end. So Taxol prevents microtubules from depolymerizing. So if you would add Taxol to the cells, the microtubules cannot shrink. And all three of these guys cause depolymerization. So when you attach these, they cause hydrolysis of the GTPs, which leads to depolymerization. So yeah, that's the one way of thinking about it, actually. Preventing the microtubule polymerizations, that means that the Taxol is causing the GTP to stay stuck onto the alpha beta tubulin dimer heterodimers, and so that causes it to continue to grow rather than depolymerize. And drugs that cause depolymerization, like these, cause hydrolysis of the GTP, which leads to catastrophe, aka depolymerization. And so here we've just got, antimitotic drugs. So microtubules play a very important role in mitosis. If you remember, they actually help a lot with like anaphase and metaphase and those phases of the cell cycle. So that's why it's important here. So you can imagine that if you prevent, if you block depolymerization or you cause depolymerization, it would mess up the cell's ability to divide and stuff like that. And that's actually the end of this lecture video. So on the longer side, I know it's like 50-something minutes, but I do hope it was helpful. We covered a lot of content in this. Again, leave comments if you have questions. I realize that I went through this stuff really quickly and that my explanations probably weren't the best, but still thought I'd get this video out as early as possible. So again, best of luck with the coming exams and the rest of the quarter. And yeah, have a good day and happy studying.